Pathogens are disease-causing organisms. In humans, they are typically bacteria and viruses, but there are also fungi, protozoa, and animal parasites. For any pathogen to infect our bodies and ultimately cause disease, there are at least 4 requirements that have to be met. In this video, we'll address those 4 requirements and take a look at how pathogens penetrate the human body and cause disease. Your objectives for this video are to define terms such as pathogen, virulence, pathogenicity, portal of entry, and portal of exit. Also, you should be able to describe how pathogens use portals of entry to enter the body and give examples. You should be able to define the terms ID50 and LD50 and explain their relationship to virulence and pathogenicity. And you should be able to describe common methods that pathogens use to adhere to host cells and hide from host immunity. Infection can be defined as the invasion of a host by microorganisms. For simplicity, in this video, I'll be talking primarily about bacteria. But remember that infections could be caused by other microbes such as viruses, protozoa, helminths, fungi, and prions as well. So during an infection, microorganisms gain entry to a host and then reproduce in or on a host's tissue. If the microbe can't be stopped by the immune system, a disease may result. Now remember that disease and infection are not the same thing. Disease is a change in the state of good health, whereas an infection is just the invasion of a host. The invasion of a host can lead to the change in good health, but they're not the same thing. Disease-causing microorganisms are called pathogens. Pathogens can be divided into 3 main groups. You have your primary or frank pathogens. These are the most probable causes of an infectious disease. For example, Borrelia burgdorferi is the cause of Lyme disease, or SARS-CoV-2 is the cause of COVID-19. Opportunistic pathogens are those pathogens or those microbes that cause disease only under specific conditions, such as when one has a weakened normal flora or is immunocompromised. The third group of microbes are the non-pathogens. These are microorganisms that rarely or never cause human disease, and truth be told, most microorganisms fall into this non-pathogen category. A host's resistance to infection and disease is due to its immunity. Resistance and immunity are the same thing. So when someone says resistance, you should think immunity, and when you hear immunity, you should think resistance. A host's resistance, aka its immunity, to infection and disease is due to a number of structures in the human body. Many of these belong to the innate immune system. Your innate immunity is nonspecific, and it's the same for everyone. This is the resistance that you were born with. Most notably, the skin and mucous membranes provide a barrier to infection, but also body substances like stomach acids, sweat, tears, complement, and defensins provide resistance as well. You might hear people call these structures and chemicals the first line of defense against infection. Your innate immunity is enhanced by elements of the adaptive immune system. Your adaptive immune system consists of phagocytic cells like your T cells and defensive proteins like antibodies. These components are formed based upon the pathogens that you come in contact with. They're educated by the pathogens that have infected you already at least once in the past, or by antigens that came in on vaccines. Because this second part of our resistance develops as we age, and because it also weakens as we age, typically the very young and the very old have somewhat lowered resistance to disease. People who are immunosuppressed or immunocompromised may also be more susceptible to infection and disease, but usually this is because they're on therapies or they have a disease that reduces the number and effectiveness of your adaptive immunity components. Pathogenicity is the ability of a microorganism, like a bacterium, to cause disease. Another way of thinking about it is that pathogenicity is the ability of a microorganism to invade a host, cause disease, and evade or escape host defenses. The more pathogenic a microbe is, typically the more virulent it is. Virulence is the extent to which a microbe can infect and harm a host. Virulence factors are structures and substances that help microorganisms accomplish this. Some virulence factors that you've already heard of include capsules and toxins, but there are others like adherence factors and siderophores, which we'll look at later. Once a microorganism infects a host, it can cause harm to that host in a couple of different ways. It might directly damage host tissues by producing a toxin that causes cells to lyse, for example. Or it might cause harm to a host by indirectly killing cells. Basically, as the microorganism reproduces itself, it produces perhaps unusual lipids, or it produces lots and lots of proteins. And these things can poison a cell and cause the cell to undergo apoptosis, a form of cellular suicide or cell death. In order for a microorganism to start an infection or cause a disease, it has to gain entry to the human body first. Pathogens enter the human body via portals of entry. Portals of entry are the sites that pathogens use to enter a host's body and start an infection or cause a disease. The most common portal of entry is the mucous membranes of the respiratory system or the respiratory tract. Here, microorganisms that are inhaled on droplet nuclei, large moisture droplets, or dust come in contact with the respiratory epithelium, and the bacteria or viruses or fungi attach to those epithelial cells and they start to grow. Some examples of diseases that are caused by or that are caused via this portal of entry include the common cold, pneumonia, tuberculosis, influenza, whooping cough, measles, etc. The second most common portal of entry is the gastrointestinal tract. Here, microorganisms in food that we eat or in things that we drink, or sometimes on contaminated fingers, gain entry to the mucous membranes that line our digestive tract. And then they can attach and grow. Now, most microbes that come into our body via the gastrointestinal tract will be destroyed by stomach, like our gastric juices in our stomach, or by the enzymes that get added to the chyme in our intestines. But if the number of microbes is high enough, and if there's enough buffering of the pH in our gastric juices or in our intestinal juices, there's enough buffering so that the material isn't too acidic or too alkaline, then some microbes might survive. Examples of diseases that occur via microbes coming in contact with the mucous membranes of our intestines include hepatitis, typhoid, shigellosis, and cholera. The mucous membranes of the genital urinary tract are another portal of entry. Sexual activity, cuts, and abrasions can damage these delicate mucous membranes and provide a way in for pathogens. Bacteria and viruses such as HIV, human papillomavirus, and chlamydia bacterium use this portal of entry to cause an infection that may progress to disease. The conjunctiva is a thin mucous membrane that covers the front of the eye and lines the inside of the eyelids. It is also a portal of entry for some bacteria and viruses. Infection may result in a familiar inflammatory response that leads to reddening of the eyes as well as pain. We call this disorder, this infection, conjunctivitis or pink eye. The cutaneous membrane, aka the skin's main job, is to provide an impenetrable barrier that separates the inside of the body from the outside. Even still, some bacteria like Propionibacterium acnes gain entry to the body via pores and hair follicles. Infections such as acne and boils result when the skin acts as a portal of entry. Finally, we have the parenteral route. Here, microorganisms are deposited directly into the tissues beneath the skin or mucous membranes. And as you could probably guess, this usually happens via punctures. So if you think about Sporthrix schinkii, back in chapter 12, remember it made the nodules that look like spider bites? That fungus enters the body via the parenteral route. Bites from people or animals can also, or insects for that matter, can also be an example of entry via the parenteral route. Cuts, wounds, surgery, splitting of the skin, swellings, all of those can lead to the entry of microorganisms below the skin. So they would be examples of diseases caused via the parenteral route. Now, many microorganisms have a preferred portal of entry that they must enter to cause an infection. If they enter through a different route, they typically don't cause disease. This table is a list from your textbook of examples of pathogens that have a preferred portal of entry along with their incubation periods. Streptococcus pneumoniae is a bacterium that's inhaled. Its preferred portal of entry is through the respiratory tract. And when the bacterium comes in that way, it can cause pneumonia. If, however, this bacterium is swallowed, it causes little, if any, illness at all. That said, there are some pathogens like Yersinia pestis, the microorganism that causes the plague, and Bacillus anthracis, the causative agent of anthrax, that can actually cause disease from more than one portal of entry. So you might remember that the plague, for example, there were 3 different types. You had the Black Plague, you had the pneumonic plague, and you had the, what was the third one? Bubonic plague. So all 3 of those were caused by the bacterium coming into the body through some different portal of entry. In addition to how pathogens enter a host, the number of pathogens is important as well. The virulence of a microbe can be determined experimentally by inoculating laboratory animals with different amounts of microorganisms and observing the animals for signs of infection or disease or death over a specified period of time. The inoculum needed to cause an infection in 50% of test organisms is called the ID50 or the infectious dose 50, and it allows us to compare virulence of different microbes or the same microbe via different portals of entry under experimental conditions. In the example that you see on this slide, we're measuring the ID50 of Bacillus anthracis via 3 different portals of entry: the skin, inhalation, and ingestion. Skin, by the way, is the preferred portal of entry for Bacillus anthracis. We can see that only 10 to 50 endospores are necessary to cause infection in 50% of test animals via the skin. Inhalation is next. We need 10,000 to 20,000 endospores to cause infection in 50% of test animals. And then you have ingestion, where 250 to 1 million endospores are needed to cause an infection. What does this tell us? It tells us that this particular bacterium is more virulent when it enters the body via the portal of entry of the skin versus when it enters via inhalation or ingestion. But we can see that it still does cause infection and disease, just we need a lot more organisms in that instance. The LD50 or lethal dose 50 is a way of comparing the potency of toxins experimentally. So the lethal dose 50 of a toxin is the dose of a toxin necessary to cause death in 50% of a test population of animals. So we're measuring comparative potency. As you can see from this example, botulinum, which is the toxin produced by Clostridium tetani, has an LD50 of 0.03 nanograms per kilogram. Shiga toxin, which is a toxin produced by Shigella bacteria and by some E. coli, has an LD50 of 250 nanograms per kilogram. And staphylococcal enterotoxin is a toxin that's made by Staph aureus. You need 1,350 nanograms per kilogram. We can see by this measure that the botulinum toxin is the most potent of all of these toxins because we need such a tiny minuscule amount of toxin per kilogram in order to cause death in 50% of laboratory animals. So that's the LD50. By the way, botulinum toxin is the most potent toxin known to man. In order for microorganisms to cause disease, they have to be able to stick to a host tissue. This usually happens at a portal of entry. Let me give you an example. Think about the most common portal of entry, the mucous membranes of the respiratory system. By now, you know that epithelial tissues have mucus-producing cells and ciliated cells. The mucus-producing cells produce mucus, which coats the surface of the epithelium and traps dust and microbes that may come in on the air that we breathe. Then the ciliated cells beat that mucus up and out of the body. It beats it up towards our throat where we either cough it out or we swallow it. If a bacterium wants to infect this tissue, or a virus for that matter, it has to be able to hold on to the epithelial cells and not get washed away by mucus. In addition, the cells in the respiratory tract also reproduce quickly. So if a microbe wants to stay there, it has to reproduce at a faster rate than that of the epithelial cells. Microorganisms like bacteria have glycoproteins or lipoproteins called adhesins or ligands on their surface. These molecules bind carbohydrates attached to the surface of host cells and bring bacteria close to the surface of the cell. Think of Velcro. You know how one side of Velcro is hard plastic hooks and the other side is soft fuzzy fibers? When you bring them together, the hooks attach to the fibers and you can stick two things together? That's what we're talking about. Microbes have one part of the Velcro and hosts have the other. Now, in addition to adhesins, cells have other structures that help them stick to the surface of a host cell. For example, you have the glycocalyx. Streptococcus mutans is one of the microorganisms that that sticks to our teeth. It makes a thick glycocalyx that acts like a glue, adhering the bacteria to our teeth. Then when we eat, they digest some of the sugar from our food and they release acids which basically break down the enamel on our teeth, causing cavities. E. coli bacteria use their fimbriae to stick to the cells of our upper intestinal tract, where they damage tissues and cause unpleasant side effects like diarrhea, cramping, and gas. Some bacteria like Streptococcus pyogenes have an M protein, which we'll talk about shortly, that helps them also stick to the surface of cells in our body. If a bacterial cell is able to enter the body and stick to a tissue, the human body will mount a response to fight the invader. Now would be a good time to pause this video and watch the video The Immune System Explained Part 1 Bacterial Infection to become familiar with how the body fights back. This video is in the Lecture Videos folder for Chapter 15. Here is the link in case you want to try to go directly there. So I want you to know that there's a mini war going on in your body. It's completely normal. First, you have the microbes that colonize your body, which we've called the normal flora, and they're in competition with each other for resources like nutrients, oxygen, environments to attach to and grow in, water, things like that. The competition among the members of your normal flora keeps the microbes under control so that no one population grows and blooms and causes infection or disease. In addition to that, you have the immune system. So the immune system monitors these microbes and prevents them from spreading to tissues where they don't belong and they might cause disease. Then you have pathogens on top of this. Pathogens are disease-causing microbes that want to invade our bodies. They have to get past our natural resistance factors first, so they have to get past the skin and chemicals and things like that. They have to do it in great enough numbers, and they have to attach to a tissue. Now, the normal flora helps us because many microbes bond to the same receptors as pathogens, and they form a protective skin. So let me say that again. The normal flora helps us because many microbes in the normal flora bond to the same receptors as pathogens, and they form a protective skin. So if a pathogen makes it past our resistance and makes it past the normal flora, an infection or disease might result. Now, if you're a pathogen, you have three main goals. The first goal is to get nutrients from your host, even if it means damaging the host to release those nutrients. The second goal is to reproduce and grow in number. And the third goal is to not be detected and destroyed by the immune system while you do the first two things. Pathogens have evolved a number of mechanisms for hiding from host defenses and penetrating deeper into host tissues. Over the next few slides, I'm going to describe a handful of them. First up, we have bacterial capsules. You'll hopefully remember these from Chapter 4. Capsules, like this one, are thick layers of organized glycocalyx that are firmly attached to the cell wall. Bacteria can control the thickness of their glycocalyx, so when they're in environments like the blood, where there's lots of phagocytic cells and antibodies, they make a thick glycocalyx that masks or hides their antigens and prevent the phagocytic cells and antibodies from quote-unquote seeing the bacteria. In fact, some bacteria like Streptococcus pneumoniae and N. meningitis make capsules that contain molecules that actually mimic host molecules like sialic acid. It's as if they wrap themselves up with these host molecules so that they look like host cells. Another benefit of having a thick capsule is that if a bacterium does happen to get phagocytized, the capsule can actually prevent it from being killed. Thus, the bacterium can actually survive inside of the phagocytic cell. A third benefit of capsules is they prevent the activation of complement. What's that? You haven't heard of complement? Complement is a set of proteins produced by the liver that assemble on the surface of bacteria and punch holes in the bacterial cell, destroying them. A thick capsule prevents these proteins from attaching and activating. Many studies have shown that encapsulated strains of bacteria, like the pneumococci and like Bacillus anthracis, are more virulent and more resistant to phagocytosis and intracellular killing than are the non-encapsulated strains. Like capsules, M protein and OPA are virulence factors found on the surface of some bacteria. M protein is an adhesin found on the surface of Fimbriae for your group A streptococcal bacteria, like Streptococcus pyogenes. In addition to helping this bacterium stick to and persist on epithelial cells, M protein is essential in providing protection against phagocytosis. In fact, these cells are only phagocytized when antibodies against M protein are present or when bacteria develop mutations that inactivate M protein synthesis. Attachment of Neisseria gonorrhoeae and Neisseria meningitidis is in part mediated by the OPA proteins. When Neisseria bacteria come into a human body, initial attachment is controlled through pili. As pili depolymerize or retract, the bacterial cell is brought closer to the host epithelium. Then OPA proteins, which are in the outer membrane of the cells, glue the cells to the host epithelium, increasing their persistence and possibly allowing their invasion of the host cell. The OPA proteins also appear to inhibit T lymphocyte activation and proliferation. It also seems to inhibit antibody production in your B cells and inflammatory responses by infected epithelia. As if that wasn't enough, it also seems like OPA increases the resistance of these bacteria to killing by the complement system. Last on this slide we have mycolic acid, and by now we're all familiar with it. Remember, this is a thick waxy lipid that is embedded in the cell walls, the outer membrane. Not the outer membrane, it's embedded in the cell wall, the peptidoglycan layer of bacteria. And only a few genera of bacteria, including your mycobacterium, have mycolic acid. The hydrophobic nature of this lipid make mycobacteria resistant to antiseptics and disinfectants, as these chemicals beat up and roll off of the cells like rain on a freshly waxed car. In addition, however, if mycobacteria happen to get phagocytized by host phagocytes, mycolic acid helps prevent digestion of the bacterium and destruction. These bacteria are actually able to survive inside of host cells, making them invisible to the immune system because of mycolic acid. So in this video, I've given you some vocabulary terms. We've talked about pathogens as being disease-causing organisms. We've talked about what virulence is, pathogenicity. We've also talked about the portal of entry. What I didn't talk about was the portal of exit. The portal of exit is the site that microorganisms use to actually exit an infected host, and very often the portal of exit is the same as the portal of entry. I've also described how pathogens use portals of entry to enter the body and given some examples. I've talked about ID50 and LD50. I've explained that both of these terms come from laboratory experiments. The infectious dose 50 tells us how many microorganisms must get into a potential susceptible host in order to cause infection in 50% of those hosts. And the LD50 was the lethal dose 50, and it allows us to compare the potency of toxins. The lower the ID50 is, the more virulent a microorganism is. The lower the LD50 is, the more virulent a microorganism is. In both instances, the pathogenicity of the microorganism is also very high. Finally, I talked about common methods that pathogens use to adhere to host cells. We talked about the adhesins and glycocalyx. I also mentioned pili. And we talked a little bit about how microorganisms use capsules and proteins like M protein and OPA to avoid different parts of host immunity. I hope this video was helpful. In the next video, we'll talk about what happens once microorganisms are actually in the body and how they can avoid more host defenses. I look forward to seeing you in that video. As always, if you have any questions about anything I've talked about in this video, please don't hesitate to send me an email or give me a call.